All right. Thank you, Ashley and Jeffrey, for introducing everybody to the meeting. I know many people have come to this meeting before. It's sort of a thing that sort of like an infection of some kind. Can't really get rid of it. For those people who haven't been here before, and for those who have been here and maybe had a little too much of the wine or something else, I'm tasked with getting you sort of warmed up for the ambiance of the meeting here. And I know it's late, and many people came from eastern regions where it seems even later to them. So I'm going to try not to drag this on too long. So you may, I assume you know that this is the real title of the meeting, The World According to Jeffrey Miller. And when you tell people this, the first thing they do is they say, who is Jeffrey Miller? And so how do you figure that out, right? If you go to Google and you search, there's actually a Wikipedia entry. It's for him. He was actually one of the people killed in Kent State, Ohio, during the Kent State shootings. Jeffrey Glenn Miller, wrong initials, I guess, here. But if someone just told you this was the world according to Jeffrey Miller, this is maybe where you would end up. Fortunately, Wikipedia has a disambiguation page that points us to Jeffrey B. Miller, Jeffrey J. Miller, and Jeffrey T. Miller. And if you go to those pages, it will not make any sense. These are not people that connect somehow to the Lake Arrowhead Microbial Genomes meeting in any obvious way. I even looked at the Jeffrey with the G spelling in case there was some mistake here, but that wasn't the right person either. So who is Jeffrey Miller? Well, organizing the Lake Arrowhead Microbial Genomes meeting, you get this invitation or you hear about it somehow, and you say, I'm going to go to PubMed and search for Miller J in PubMed, and you get 13,937 publications. He's a really good scientist. Has just an amazing record of the diverse areas, diverse topics. Some a little strange, but that's okay. So clearly that is a little excessive. So you get the invitation. It's from UCLA, right? So you search for Jeffrey Miller and UCLA. So you go to Google. How can there be two Jeffrey Millers at UCLA doing microbiology? This is just brutal for people who have, trouble with name recognition and trouble with other, remembering. I have trouble remembering yesterday, let alone dealing with all the different Jeffrey Millers. So I figured, as practice, I know both of the Jeffrey Millers, of course, but as practice, I would come to the meeting and see if I could figure out which Jeffrey Miller was which Jeffrey Miller. And literally, I came in today, and what did I see? But Jeffrey Miller and Jeffrey Miller. How are you supposed to figure these things out? And we're supposed to keep track of genome sequences when we can't even keep track of who's organizing the meeting. But anyway, thank you, both Jeffrey Millers actually, for being here. And what I thought I would do is, you've heard a little bit about the meeting, but I thought I've actually been coming to the meeting since 1998. When I was a graduate student, just finishing up in graduate school, before moving to Tiger, I figured I was going to start working on microbial genomes. I should probably go to a meeting about microbial genomes. And so I came to this meeting and was just overwhelmed with the community, the sense of a community there. And it was a great step for me as a graduate student to get introduced to all the people working on, at the time it was, E. coli and a couple of other organisms, but it eventually spread into other areas. And so as part of this really love of this meeting, I've actually scanned in all of my old conference notes and the notebooks and posted them online. So there is, I think that's the one from 2000. I think 1998 is on here somewhere too. Yeah, there's 1998. I've posted PDFs of all these. Many of them I've actually done character, recognition, so converting the the scans into text. Some of these are online, but not all of them from the older meetings. I don't know if Jeffrey has all the electronic versions, but you can go to my website, tiny URL, the real Jeff Miller, and find all of these notes about the Lake Arrowhead Microbial Genomes meeting. And I've posted links to whatever I could find about the meeting. If someone else has written a blog post or the links to the websites from past meetings, I've posted them there. And I just thought I would show you, if you want to go look, there's some, obviously I'm biased because they're my notes, but there's some incredibly brilliant stuff in my notes for these meetings. So this says, I'll blow one up later, or I'd like to blow one up now. I think that referred to some sequencing machine, but I'm not sure what it was. This is really important stuff that you can find in my scans. This must have been during the baseball, run into the playoffs, so I was keeping track of my fantasy baseball team or something. I don't know what that is. This is a very important figure, genes and intergenic regions, in case you were confused. So these are some great notes from 1998 that you can go look up. All of them are there. I thought I would just show you another year. 2002, it got much, much better. I wrote down the times of all of the speakers because I think I was chairing the session and had to remember I didn't have a stopwatch. So I'm like, they started at 9:30. Now when, I got to stop them at 9:50. And so there are all these notes next to the speakers with times of the day. It's very bizarre. More, very scientific doodles. This is playing hangman, fill in the blank here, H-E blank P. There's another one, I think that's my ERA or something like that. The posters are after lunch. That's important to put in the notes. And then I don't know whose talk this was, but I'm sorry, it's not. I don't even know if it was associated with a talk or something else. Here are more of the times of the speakers later on in the session to make sure they stopped on time. But I don't know if anybody here is obsessed with history. As an evolutionary biologist, I care a lot about history, and there are lots of different ways of approaching history. I have the artifacts, which are these scans of the meeting notes, and we have reflections of the people who have been at the meeting. But as many people know, I really like quotes taken out of context as a way to represent nothing or something going on at the meetings. And so I'm going to go through some of the quotes. I write these down during all the meetings. Rarely do I have a name associated with them, although I do remember some of them, who they came from. And, it's now the era of Twitter and other things, so if you say something really bad, your name will be associated with it now. But these ones aren't exactly clear. So someone, probably Fred Blattner, was talking about the space-time continuum of genes and genomes. Microbes not only have a lot of sex, they have lots of weird sex. Gene sequences are the wormhole that allows one to tunnel into the past. This is how you do metagenomics on $50, and that's Canadian dollars. I don't know, was that Fiona? I don't remember who that was. Yeah. Very insightful things like the human guts are a real milieu of stuff. Antibiotics do not kill things, they corrupt them. And I could go, on. I actually have pages of these. I agree with this statement. I don't know when that point is, but I agree with it. This is a real quote referring to a figure where the rectal swab data was in a tan color. This must have been Fred Blattner also. I like the people who say something and then realize it really wasn't important and they try and take it back, but Lance, where are you? This will probably come up again sometime during this meeting. No idea who this is. I like predicting the future, so this is probably my favorite one. Someone really said this. All right, I mean, there are so many good quotes at this meeting. I thought this was going to go the way of like the Princess Bride, but it didn't quite go that way. I, I wrote it down. I still don't know what it means. Now, this is very relevant because I'm not going to wrap up yet, but the crickets are always chirping, and people are always getting antsy when the crickets start chirping. So, as an evolutionary biologist, quotes are, of course, not everything. So I think people, if you were here 4 years ago, you may have seen me briefly mention this. If you want to study the evolution of the Lake Arrowhead meeting, there's a way that we all can do this, which is you can make Lake Arrowhead into a peptide sequence and do a blast search, and then build an evolutionary tree of the resulting sequences that match Lake Arrowhead. I haven't done this recently. The web, I was going to do this a few minutes ago. The web connectivity wasn't fast enough for me to actually get this done and download anything. But that's the result of a Lake Arrowhead blast search from a few years ago. There were other sequences. So, for example, something from Tsiona, it sort of looks like Arrowhead, Earl-Wahe. And here's Growahead. So there is Lake Arrowhead hidden, or sort of close relatives of Lake Arrowhead hidden in many genomes. Here's, as people probably know, the only thing I know how to do is build evolutionary trees and try to interpret them. So this is the evolutionary tree of that blast search. I guess Lake Arrowhead here is an outgroup relative to many other clades. You should do this at home. When you have better web connectivity, or you can log on, you should compare Jeffrey F. Miller and Jeffrey H. Miller and maybe all the other Jeffrey Millers and see what you get. I haven't done that actually, but I'm sure it's very interesting. So another way to think about history of a meeting is to try and compare how the ideas, the topics of the meeting have changed over time. And with the record that we have of those brilliant notes that I showed you of the scans of the meeting, we can actually go back and try and understand what the meeting was like 10 years ago and 14 years ago and 2 years ago and look at how it's changed over time. So you can go back and look at the scans. We've done this in a few, tech-savvy ways, but I'm going to show you my own impressionistic view of what the history of the meeting is, which is I thought about the meeting for a few minutes over time, and then I said, what was that meeting that year about? It's I'm sure that it's very accurate here. So the first one that I went to was 1998, and as far as I remember, the entire meeting was about genome sequences. I don't think there was zero other things covered in the meeting. Of course, that's not true. But, in 2000, it was, I think about 100% about genome. Maybe this relates to the fact that I only went to the talks about genome sequences. I was working at Tiger at the time, so. And then finally, we got some change. In 2002, there was a lot of talk about genomes, but also a lot of talk about microbial communities. Mostly, ribosomal RNA surveys of microbial communities. And then in 2004, we got the combination of genomes, microbial communities, and metagenomes. Finally, we got into the meat of metagenomes in 2004. And then in 2006, the metagenomes started to sort of take over the topics. Genomes became kind of boring, a single cultured organism, who cares about that? Until eventually in 2008, the whole meeting, there was nothing about, it's called Microbial Genomes. I'm sure there was absolutely nothing about genomes in the meeting. It was all about metagenomes and microbial communities in some way. And then finally, we got the good topic in 2010. Microbiomes came along to the Lake Arrowhead meeting. There was still some metagenomic stuff of, soil and oceans and other places, but microbiomes, host-associated microbial communities came in, and in 2012, they spread. And then this year, in case you don't know, almost all of the talks are about microbiome. It's true, I swear. She's shaking her head. It's just, it's definitely true. Microbiomes are everything now, right? I mean, microbiomes are taking over the planet. So I think that if we think about the evolution of the meeting, the next meeting is going to be 100% about microbiomes. So the question is, what are those topics going to be? Where is microbiome research going? I mean, it is, as many people know, I give out these Overselling the Microbiome Award on my blog, but actually, I think microbiomes are awesome, and I'd like to give you a preview of what I'm imagining as one of the co-organizers, even if my co-organizers will let me invite some of these talks, for the 2016 meeting and beyond, predictive talks about the future. This paper was published in Biology Direct about microbiome components to religious rituals, and it did in fact talk about midichlorians. And it's really interesting, actually, not the paper itself, but the reviewers' comments on the paper, because Biology Direct publishes the peer review of the paper before it's published, and then the author's responses. You really should read this. It's completely awesome. There's some great review comments for this paper. I think Rob Knight, Eugene Koonin, and I can't remember who the other one is. And it's actually actually really kind of interesting. I'm hoping we can get Al Gore out to give a talk. He actually invented the microbiome. He did, in fact, talk in an interview about his vegan diet and how he's doing it in part to shape his microbiome. So I figure in 4 years, maybe, he will have invented all microbiome studies. Microbiomes are going to be an excuse for all sorts of issues. We've already heard a little bit about this. I think it will show up in court cases where you try and say, my microbiome made me do it, like they have here. So I think that would be a really interesting topic to have at the meeting. There's a lot of accessory, so to speak, information about microbiomes. I found this yesterday. There's a probiotic toilet paper so that you can clean out, it's not for you, yourself, but it's to shape the microbiome of your toilet. There is truly talk, and I mean, there's actually real science on this, but it's also kind of funny, on the microbiome and doping, performance enhancement associated with the microbiome. I found a truly interesting talk about this that was posted online. Could the microbiome be used as an ergogenic aid instead of blood doping? Is there going to be bacterial doping related to metabolism, overtraining, travel, and a variety of other things? The authors of this talk even proposed that as part of the biological passport, you might want to have a microbiological passport associated to prevent microbial doping. So I think that should definitely be part of this meeting next time. If people haven't seen it, there are lots of people who talk about microbiomes associated with diet. There is, in fact, a book called The Microbiome Diet. There are some real interesting microbiome-associated diet concepts out there. I don't think they're contained in this book. If you're interested in expanding the topic of microbiome beyond, sort of normal science, there's some fascinatingly bizarre and interesting stuff out there on this internet thing. This is in the wrong order, but so there's a person called Sunshine Mary who has a blog, Christian wife and mother, against feminism and female suffrage, pro-family, and she has a whole thing about how the microbiome might actually explain why we should be monogamous. When a woman comes together with her husband, she's receiving more than just semen. She is also being seeded by receiving a big dose of his microbiome inside of her. A microbiome which literally affects who she is and how she thinks and feels. In a very real sense, we become part of the men we have sexual relations with. And she goes on to then quote from various parts of the Bible about how this is relevant to the microbiome. You can't make this up, or I can't make this up. I don't know, maybe you can, but I think the title of this says it all. But this would make an awesome talk, maybe for the first night of the meeting. There's someone named Sarah Rich on Twitter who posted about this, and there literally is someone proposing sort of an art concept, but a real concept, fart alarms, where you could sense the change in the microbiome in an environment and assign that change to a particular person. There is a lot on intelligent design sites now that focus on the microbiome because, of course, the microbiome is perfectly tuned to enhance our biology, perfect mutualistic associations with all microbes that have ever touched any human being. It's on this website. It must be true. There are things that you can even shocked me, which is pretty hard. And I posted about this on Twitter earlier. There was a story in Jezebel a couple of days ago. Microbiomes want to make some part of your female anatomy taste like Diet Cola. Through microbiome engineering, there's a real discussion of how this might possibly work. It's deranged and crazy, but it could make a good talk, right? No. So I think for 2016, we have a list of amazing microbiome-related topics. I'm going to be nominating all of them for every session. And I probably won't be here again, I guess. In honesty, this meeting has been amazing for me. For many people, as Ashley and Jeffrey already said, there are incredibly diverse topics, obviously not all about the microbiome, that have happened at this meeting. But since I am standing between you and Jeffrey's free wine and the food and relaxation, I didn't want to be too serious, and I didn't want to go into scientific depth about anything. But I just want to leave you with one thought about the 2018 meeting. Because in fact, I think that with this trend of microbiomes taking over all the topics and those amazingly exciting things that we could have for 2016, I think the microbiomes will be giving the talks in 2018. So we won't be needed. And that's it.